afterward. And then when that presentation is over, I will then introduce the subsequent speaker. So without further ado, I want to introduce Dr. Franz Rosenzweig, who's going to talk about no starvation here.
This is about right? Okay, great. Okay, so I think that everybody over the last 24 hours, maybe over the last 1 hour, has enjoyed the awesome power of yeast. After all, yeast and humans and triticum and grape have been collaborating for many thousands of years to give many of the things that we most enjoy in life. And I think that in this audience of science writers, if anybody has gone to a yeast genetics or cell biology meeting, you've heard trumpeted over and over again this phrase, the awesome power of yeast genetics. And in fact, yeast have awesome power, and they have informed our views of basic phenomena such as cancer, in particular the cell cycle work that was undertaken by Lee Hartwell in the early 1970s. Much of what we understand about signal transduction is also the result of the awesome power of yeast genetics. Yeast have been used more recently to illuminate uncertain issues in evolutionary biology, such as the meaning of ploidy and the adaptive value of sex. But today we're going to be talking about how yeast genetics has informed our view of aging. And the first indication that yeast could be a model system for studying aging came from work published back in the late 1950s by Mortimer and Johnson. I see that this has gone out, so I'm just going to dispose of it. And Mortimer and Johnson made the observation that yeast have a finite lifespan just like we do. That you can take a mother cell and propagate her, dissect away her buds, and that mother cell, in fact, will have a finite lifespan. They also have chronological lifespans. And later researchers, including people like Michael Jeswinski, have gone out and demonstrated that not only do you have finite replicative and chronological lifespans, but if you get wild strains from nature, not mutants, but wild strains and such exist out there in the world, then in fact there's genetic variation for the differences in lifespan in yeast, and where you have genetic variation, therefore you have a genetic basis for this trait. And since then, there are a number of important works that have come out from the labs of David Sinclair, from the Guarente lab, from Matt Kaberlein's lab, that have identified specific aging genes. Well, what I would like to convince you today is that the power of yeast genetics to inform our view of these basic biological processes has actually been realized in relatively few contexts. And by that, I mean you either go to a yeast lab and people are growing yeast either dilution cultures up here, a batch culture where you just have a suspension of yeast cells, or they're taking these, diluting them, and putting them on agar plates and looking at their growth on solid media. Obviously, the combination of yeast mutants that are possible and the combination of different media constituents that you can put in these two different environments allows for limitless investigation of the connections between genotype and phenotype. However, there are other ways to cultivate yeast. One way that has become increasingly popular, and we introduced or reintroduced this old technique back in the late 1990s, was the use of chemostats, where you grow cells in a culture, basically planktonic, free-floating cells, but in a system devised originally back in the 40s by Jacques Monod, but in a system where you have continuous feed of some limiting nutrient, enabling you to establish a steady state within this population, and you can assay this steady state for a variety of parameters, including gene expression. So chemostats are very useful for gene expression studies in yeast and other microorganisms. Well, a few years back, actually about 3 years to be exact, I became aware, by virtue of just giving a talk at another institution, that there is another way to grow microbes, another way in which yeast can be propagated, and that is through the use of so-called immobilized cell reactors. Now, immobilized cell reactors have been the province to date of chemical engineers, people in biotech, and basically what goes on here is something that's qualitatively different from these other modes of culture. Cells are immobilized in a semi-solid matrix, and they are formed basically in these small tapioca-sized beads, and these beads then are loaded into a bioreactor which can be continuously or discontinuously fed. The interesting thing that was pointed out to me at this meeting, which was at Utah State University, where I met some very talented chemical engineers, the interesting thing is that whereas you had to maintain steady state here by putting in a limiting amount of nutrient, you could feed these cells very rich media and you appeared to have a stable system. So, just as a quick review, because I'm going to be talking about these different culture states, and what I'm going to be promoting is the idea that you can use this last culture state on which no gene expression studies have been done to date using immobilized cell reactors, that you can use this as a powerful system to study yeast aging in a very different, and I would suggest to you, important context. So, to review, agar plate, growth on solid media, the nutrients are eventually depleted. It's a closed system. Batch culture, also a closed system, the nutrients are eventually depleted. In a chemostat, you have an open system where you have continuous feed of a limiting nutrient so that you have cells reproducing, but reproducing at a slow rate. Basically, the rate at which you're delivering media into the chemostat vessel. So you can tune growth rate basically by tuning the dilution rate through the pump. In the immobilized cell reactor, we also have an open system where you have continuous feed, but it's possible to feed these cells continuously excess nutrients beyond which they need for their common growth and metabolism. And so, the topic then today is longevity in the absence of caloric restriction. And the question then is, do immobilized cells age? For those literary-minded in the audience, the alternative title here is Das Hefeperlenspiel. I'll give you guys a quiz on that at the end of the talk. So, a bit of background on cell immobilization, on the questions that motivated our study, and then our experimental results to date, and then summary and future directions, the directions that the NIH has recently funded us to pursue. All right. So, we immobilize our cells in alginate, and alginate is obtained from algae. And so the commercial source is usually giant kelp. This is what the structural polymer looks like. And when you crystallize this, you can get what this sort of powder-looking substance, sodium alginate, that you can buy off the shelf either from Fisher VWR. You can get it online. In fact, if you Google alginate, I guarantee you'll come up with some very odd articles and images. People use it for life casting. And I'll leave you with that keyword if you want to explore that odd literature. What you can do, instead of making life casts of naked people, you can form these little beads. And these little beads are formed in a fashion that I'll show you in just a moment. But the point is that you can take these beads and impregnate them with yeast and study them then in a very different context from these other 3 types of culture conditions that I've described a moment ago. All right, so how do we do it? You take a suspension of yeast, and in all the experiments that I'll be describing today, we're using the common wild-type strain S288C. An important point, we're using a haploid strain of yeast. And you take a suspension of sodium alginate, you pump them together, and you put them drop-wise into a solution of calcium chloride. When you do this, the negative side groups on the structural polymer are cross-linked by the calcium, and these cross-links form a gel. This is a small test tube. This is the size of one of the beads that you create this way. They're a couple of millimeters in diameter. So important things to consider are that the bead size, to a certain extent, the geometry of these beads, as well as their hardness and the number of cells that you put into them, can be controlled experimentally. And for our purposes, it's extremely important that these beads can be reversibly dissolved by a process that chemists call reversible inotropic gelation. Shakespeare would have loved that. But the point is that you can put a chelating agent like EDTA or sodium metaphosphate onto these beads. They'll break apart, and you can take the cells out. It does not affect their viability one iota. The immobilized cell reactor that we use, I'd like to emphasize that this is not our invention, and it's not even the invention of our collaborators. People have been using immobilized cell reactors in biotechnology for over 20 years. The one that we use is configured like this. You have this contraption. It's autoclavable. It's water jacketed for constant temperature. We run these experiments at 32°C. And our chemical engineering colleagues have optimized the bead and the column geometries in order to assure that the system is not diffusion limited, but rather reaction limited. It's limited by the reaction capacity of the enzymes within these cells. Now, a couple of important features to note is that the void volume, in other words, this is not how much these guys are urinating, but rather it is the volume between the beads within the column. This void volume is exchanged on average of once every minute, and the nutrients are being constantly replenished, and the cells are seeing up to 10 grams per liter glucose. That's 10% glucose. This is what you would get if you went to have a glucose tolerance test done at your physician's. So there's plenty of nutrients. We are replenishing not just glucose, but all other essential nutrients on a daily or every other day basis. Cells that break through to the surface of the bead and enter the planktonic state are removed from the stream, and then the ethanol is removed in order to avoid any possibility of product inhibition, and then the pH and temperature of this much larger reservoir of the feed is automatically controlled. All right. So much of the talk is going to be focused on a couple of words. And the words are that the yeasts that we're studying behave differently and profoundly differently when they're immobilized than when they are planktonic. And although there's been some really exquisite physiological work done on cell immobilization, to date no one has done gene expression studies, and no one has done very much in terms of testing different yeast mutants in the immobilized state to see how that affects their physiological performance, much less their life history and demographic parameters. So comparing immobilized cultures that are continuously fed rich media with batch cultures at mid-log phase when they're growing the fastest, a few things become apparent even in a short-term experiment. First of all, the yield of ethanol is up in the immobilized cells, although by only 5 to 10%, but it's highly reproducible. More dramatic is that the yield of biomass is down by several-fold. And as a consequence, if you normalize the ethanol production rate by this diminished biomass so that you have cell-specific or biomass-specific rates of ethanol production, what you have in the immobilized cell reactors are reaction rates that are 4.5 times higher in our experiments, and reports in the older literature of 10-fold or greater rates of ethanol production. Consistent, or I wouldn't say consistent, but concomitant with these physiological changes, there are reports in the chemical engineering literature that yeast become highly stress tolerant in prolonged immobilized batch culture, meaning they're resistant to heat, they're resistant to ethanol, they're resistant to organic solvents. But what really piqued my interest when I first learned about this other state of being, potentially, for yeast, is that they might have longer lifespan. And there was a report in the literature over 25 years ago from a USDA lab that these very high rates of metabolic activity could be sustained in cultures that had limited or no growth for up to 90 days. This was extraordinary in consideration of the fact that since Mortimer and other people have been studying aging in yeast, that yeast are not known to have lifespans that exceed a few days to, at most, a very few weeks. All right. So aging in yeast, as I alluded to in my introductory remarks, can be viewed in two ways. It can be viewed in terms of replicative lifespan, and this is the number of mitotic divisions that a mother cell will undergo and the observation that she senesces as she undergoes these successive mitotic divisions. Another way in which yeast aging be studied is in terms of the chronological lifespan, and that's basically the number of days that a non-dividing cell can spend in stationary phase. So, just to summarize, replicative aging, how a mother cell senesces; chronological, how a non-dividing mother cell senesces over time. In yeast, necessarily, because to date it has not been possible to uncouple metabolism from reproduction, caloric restriction has been the name of the game. So all chronological aging studies in yeast have been conducted under calorically restricted conditions, basically trying to answer the question, how long can a non-dividing cell live basically under starvation conditions, right? And this has not, I'm not taking issue with this, many of these excellent studies, it's just that I think that there's something missing in the puzzle. Well, caloric restriction, as you almost certainly all know, has been shown to extend longevity in a number of species up and down the evolutionary ladder, if you believe in such things. Not in evolution, the evolutionary ladder. Let me emphasize that, please. Let me say that again. Evolution and evolutionary ladder. That in Drosophila, in C. elegans, in yeast, in mouse. It was discovered 75 years ago in rat. And of course, there was that great study that was published this past summer showing that caloric restriction also works in primates. So how does it work? That is a matter of some contention. I would refer you to an excellent review of this by David Sinclair in Mechanisms of Aging and Development. And this is a summary of his take on this, and that is that the evidence that caloric restriction delays development or somehow reduces metabolic rate is weak at best. So he is dismissive, and I agree with him. He's dismissive of those arguments. But what is unarguable about caloric restriction is that it alters and attenuates the glucocorticoid cascade. It reduces fat, not surprisingly. It reduces the load of reactive oxygen species that organisms have to bear, and it reduces overall the levels of apoptosis, or programmed cell death. The hypothesis that he advances in this paper, which has gained considerable attention since then and existed in various forms before, is the Hormesis Hypothesis. And that is the view that a low-intensity stress, in fact, may be protective because it elicits other stress responses. Okay, so as Shakespeare would put it, you know, sweet are the uses of adversity. How does it go? Like the toad, ugly and infectious, which yet a jewel wears on its head. And that's all you get of that today. All right, so aging in yeast. So we've talked about chronological lifespan and replicative lifespan. Hereafter, we're going to focus on chronological aging. So starved yeast has been used as a model to study the aging of post-mitotic cells in higher eukaryotes. And in fact, as was the case with the RAS genes, that they're mammalian homologs of some of the genes that have been uncovered in these screens for chronological longevity genes. Not surprisingly, those loci found to extend yeast lifespan under caloric restriction tend to fall into the categories of stress response and or starvation response. But as all of you guys know, many post-mitotic, terminally differentiated cells, for example, neurons, are metabolically active, very long-lived, and experience limited caloric restriction because most animals will go to great lengths to protect the central nervous system and buffer it against sort of the vagaries of nutritional status. And so our goal then is to develop a system using the awesome power of yeast genetics and uncouple reproduction from metabolism. And if we're able to do this, then our expectation is that we should be able to discover new categories of longevity genes. So the questions that we're seeking to address are, first of all, how does the demography and physiology of immobilized, or these encapsulated cells, differ from that of planktonic, or the free-floating cells that are most commonly studied in yeast biology? What gene expression program enables yeast apparently to minimize or cease reproduction and jack up metabolic rate? So we saw that these yeast are capable of throughputing glucose at levels that are 5 to 10-fold higher than cells in mid-log phase batch culture. And then, of course, we want to see whether planktonic and immobilized yeast age differently and seek to identify caloric restriction independent yeast genes. So the experimental design is as follows. The treatments that we use, we're comparing planktonic batch, planktonic chemostat cells, the immobilized cells, and because these immobilized cells that are being fed all of this glucose evolve so much carbon dioxide, they basically are scrubbing all of the oxygen out of the system. Perforce, these are anaerobic cultures. And so we did, for comparison, our batch and chemostat cultures under anaerobic conditions. However, because much of the aging literature in yeast has been done under aerobic conditions, we carried along planktonic batch culture experiments also under these conditions in order to compare to values others have obtained and reported in the literature. So we're interested in things like demography, population size, age structure. We're interested in physiology, and in particular, what these yeast are doing in terms of their onboard carbohydrate reserves. And we're interested in their genomics, not only their transcription profile, but also their DNA content and their DNA integrity, which reveals, as I will show you momentarily, a lot about what's going on with respect to the cell cycle in these cell populations. All right, just to remind you, we're going to be comparing the immobilized system where we have excess nutrients being continuously fed to the closed system of planktonic batch and the open system of planktonic chemostats. So here's a short-term culture, which is sort of characteristic of these types of experiments. Going from the bottom up, planktonic anaerobic chemostats you inoculate, and you quickly reach stationary, not a stationary phase, but rather a steady state, where the amount of growth is being balanced by the cells simply being washed out of the system, okay? If you go the next up, planktonic anaerobic batch, here you have the typical log phase going to stationary phase, and cells cease to grow because they've depleted the media of all available nutrients. And the last are the immobilized cells, where that's in the solid circle, or excuse me, this one right here. You have an initial phase of population expansion, and then cells reach a point where apparently you have no continued growth in spite of the fact that you have an abundance of nutrients being fed these cells. All right, we would be interested in their reproductive status and their replicative age, so comparing budding index, basically just looking under the scope and seeing how many cells have produced a bud. At the first day, both planktonic and immobilized cells show a budding pattern characteristic of a population that's expanding rapidly. After 5 days, the planktonic cells have reduced in the number of cells that are budded, but even more so is this true of the immobilized cell population, where less than 10% of the cells after 5 days are budded. Now, you can determine the age structure of a yeast population, or at least the replicative age, by counting the number of bud scars. These are basically the residues of a past budding event that can be stained using calcifluor and looked at under epifluorescence microscopy. And what's remarkable is that going from day one population, remember this is going You're going to have several rounds of division before they cease dividing. Day 5, you have a population of cells that is predominantly unbudded and in which you find a few members of these older replicative age classes. So by day 5 in the immobilized cultures, you have a large population of unbudded cells. Well, this is kind of where the rubber meets the road about cell survivorship. Here we are comparing immobilized anaerobic cells in the dotted lines, solid circles, to the planktonic anaerobic cultures. And you see comparing survivorship over time, that you have a rapid decline in survivorship in these cells that are calorically restricted compared to these cells that are non-calorically restricted. If you do the experiment under aerobic conditions, as most people do, all it does is push the survivorship curve over to the right. Now, one thing that has been associated with prolonged longevity in yeast has been their ability to accumulate glycogen. Glycogen in yeast, like in ourselves, is one of the major storage carbohydrates, and high glycogen levels have been associated with the ability to live a long time in stationary phase. So here are the levels of this storage carbohydrate glycogen comparing to planktonic anaerobic culture. Here are the immobilized cells, and here are the aerobic planktonic cells. And what you can see is that you have the immobilized cell hyperaccumulating this key storage carbohydrate, but the planktonic cells not. So basically, one thing they're doing with all this glucose is just socking it away, perhaps for a rainy day. Okay, so I'm making the point that we have, we're trying to uncouple metabolism from reproduction. And if indeed done that, then there should be hallmark features visible both in terms of gene expression as well as in the profile of DNA content in cells that would suggest that the cell cycle has arrested. Just as a Bio 101 review, this is the eukaryotic cell cycle that enables cells to realize their dream of becoming 2. Here's G1, DNA synthesis, gap 2, mitosis, cytokinesis, and the 2 cells. It turns out that if you stain the DNA, you are able to follow the cell cycle, and in fact you can follow apoptosis because as cells break down, you tend to have a shoulder over here. Here you have an active population of cells where G1 and G2 are these 2 peaks. If you do this in diploids, you have G1 and G2. If you look at triploids, G1 and G2. So you can follow the cell cycle to an approximation by doing fluorescence-activated cell sorting of cells that have been stained with a stain specific for DNA. So here are planktonic cells, and what you can see going from day 1 to day 5 to day 10 to day 17 is just as we saw in our index, where you had a low level of mitotic activity even in the calorically restricted cells, you appear to have a low level of activity also in these cells that are subjected to prolonged nutrient deprivation. Moreover, you have the accumulation of signal over here indicative perhaps of apoptosis. Compare this then to the immobilized cells where you have the G1 and G2 peaks first apparent on day 1 but not visible thereafter. And aside from this blip that we have yet satisfactorily to explain, most of the cell population appears to be solidly arrested in G1, suggesting that we have achieved our goal of uncoupling metabolism from reproduction in this model eukaryote. So now we wanted to move on to trying to understand the genetic basis, the genetic program that underlies this unusual state. And so we conducted a set of microarray experiments using Affymatrix microarrays. The methods paper on this is out, Krukeberg et al. 2009. The other results that I'll report are in review or in prep, and I would ask you kindly to consult me before you publish these, and let me give you an update on the status of their peer review. So the the systems that we're comparing, again, are immobilized open, planktonic batch closed, and then these chemostat-grown cells. And for the cells that we're looking at in batch culture, we're looking at the planktonic log phase, planktonic stationary phase, and then we're looking at the chemostat-grown cells. All right, and I'll just say as an aside, there are good reasons why people have not done expression studies on immobilized cells. The technique for getting RNA out of these damn things is really a pain in the ass. Yeah, you ask me over dinner, and I'll tell you what my postdoc ended up having to do. Or you can read this paper and laugh. You know, it's pretty comical. All right, so here's a heat map of expression data for 5,700 genes. And this is going to be a consistent way of presentation where the planktonic time course is over here. Batch log, batch stationary, and chemostat cells are there. And so planktonic is always going to be that. I feel like I'm doing the hokey pokey up here. And the immobilized cells are over there. So a few things to note. Each of the columns represents at least 2, and in most cases 4, biological replicates. The beads are made with stationary phase cells, and therefore it's no accident that batch stationary and day 1 look very much like one another. You have this transition in gene expression profile going from day 0 to day 3. And aside from some genes that were oddly upregulated or downregulated in day 10, you have a fairly stable pattern of gene expression in these cells being fed massive amounts of glucose. And in fact, going from day 3 to day 17, only about 100 or so are up by or down by the twofold criterion. So what are these genes? Well, the expression of many genes with hypothetical or unknown function are altered in the immobilized state. And in fact, this category of genes is overrepresented in the gene set that we discovered. Global regulators, and there's not going to be a test on this. The point is that many genes that regulate other genes are differentially expressed and in combinations that have not ever been discovered before in other types of growth conditions, suggesting, to me at least, that there are novel regulatory pathways to be discovered in yeast. I mean, every year at the yeast meetings, we think that we've gotten closer to understanding everything, and yet, just a little bit further, and it seems like we have, decades and decades more of work to do. So this is actually sort of a cool thing. Glycolysis, as you might expect, here we're averaging the day 3 to day 17 data, where red is immobilized, up by twofold or more, green down by twofold or more. These are glycolytically active cells, and it's not surprising that this is compared even to mid-log phase cells that are also metabolically active. So the committed steps in glycolysis of hexokinase, phosphofructokinase, PGK are up manyfold over the highest expression that is observed in these planktonic cells. The one exception to this, incidentally, pyruvate kinase, is also a cell division cycle gene. Actually, I did my thesis on PK, and it's a great gene, having this dual function, both in catabolism as well as in regulating the cell cycle. All right, so under conditions, you have plenty of glucose, excuse me, we have low glucose and oxygen tension. Let me take a step back. Under conditions where you have low glucose and high oxygen tension, the TCA cycle is kicked in, but in the immobilized cell reactors, we have the opposite conditions, and so the TCA cycle is downregulated, and rather conspicuously so in the sense that citrate synthase and aconitase, the two rate-limiting initial steps in the TCA cycle, barely detectable levels of gene expression. So we're going to focus much of the rest of the talk on the cell cycle, because indeed if these cells are G1 arrested, then they must be doing odd things. And they're doing very odd things considering the fact that they're seeing plenty of nutrients, and yet they're acting kind of like stationary phase cells. So I would pay particular attention to the commitment to undergo DNA replication, START, which includes regulatory cyclins as well as other genes like CDC28 and 485. So again, you're not going to be quizzed on this massive cell division cycle map, but just to the eye, it should be obvious that many of the important players like CDC8, PICL1, CLIN1, CDC28 again, many of the key regulatory genes that enable cells to either commit to going into the S phase or to escape from the S phase and go into G2 are conspicuously downregulated. And so basically the blocks appear to be at both of these places, and it may be due in part to the fact that genes like CDC28 play important roles in both of these processes, and CDC28 and the CLINs are essentially off, even though these cells are being fed abundant amounts of glucose. Well, yeast not only do asexual reproduction, and not only can they be propagated as we have as haploids or as diploids, but they also undergo meiosis, and undergoing meiosis, they form spores. And so the meiosis, as a reminder, is one round of DNA replication, 2 rounds of cell division, and in yeast, sporulation is a generic response to low nutrients. And it yields dormant, keyword, stress-resistant haploid ascospores. So how does the expression profile of this experimental yeast, which is a wild-type haploid, what does it look like in the immobilized state? Very different from the planktonic cells. Again, just pay attention to what's in blue. These are meiosis-specific proteins required for meiotic recombination, required or highly induced during sporulation, required for dissociation of chromosomes from the mitotic spindle, etc. Many of the genes that are highly expressed in sporulating cells are also highly expressed in these cells that are G1 arrested but metabolically active. So diploid cells are the cells that undergo meiosis, but yeast in these experiments are haploid. Well, it turns out that many of the genes involved in cell wall remodeling are also differentially expressed. And this comes possibly not as a surprise, because one characteristic feature of sporulation is remodeling of the cell wall to make these highly resistant dormant ascospores. And again, here you have the various planktonic states on which we've done gene expression profiling, and here you have the immobilized cells. This is just the day 0, which is basically stationary phase yeast. And all of these genes involved in spore wall synthesis and basically remodeling the cell wall by tearing certain things down and building other things up, these remodeling genes are conspicuously highly expressed in these cells. So many of these genes in meiosis, many of these genes in sporulation are considered stress response genes responding to low nutrients, basically poor growth conditions. All right, so we've been waiting to see now what chronological aging genes are doing. And here are some of the key players that have been identified over the last decade by exhaustive screens, genes like REM15, MSN4, SCP1, and the aconitases, and you see that they are differentially expressed with respect to any of the planktonic states. And in particular, I would call your attention to REM15, which is turning out to be a central regulator of metabolism as well as the cell cycle in yeast. It is a glucose repressible protein kinase. It is usually repressed by abundant glucose, but it is highly expressed. Bearing in mind, this looks black here, but bear in mind that what we're comparing it to are these very deeply green squares for chemostat batch cultured cells. MSN4 is also highly increased in its expression. So, given that there's observations in the very recent literature, a great paper out of the Longo and Fabrizio lab in last year's PLOS Genetics, that REM15 may play a central role in mediating calorically restricted longevity in yeast, my postdoc decided to do the simple experiment of comparing how the cell cycle looks in planktonic cells to how the cell cycle looks if you do the experiment with a REM15 deletant strain. And so, as you saw before, you start out with a mitotically active population. This G2 peak diminishes quickly as the immobilization experiment progresses, and then compare that to REM15, where you have this persistent G2 peak indicating that cell division continues in the immobilized state, whereas you had cell arrest over in the wild type. So how might this be working? So I would refer you to these two papers, Feng et al. From PLOS One and Wee et al. From PLOS Genetics. How might REM15 be acting? Well, the conventional wisdom now is that REM15 responds to a variety of different nutrient signaling pathways, the TOR, the SCH pathway, glucose and phosphate. So changes in ambient nutrients basically will result in the activation of this pathway, and then REM15 in turn activates MSN2 and MSN4. These are transcription factors that then migrate from the cytoplasm into the nucleus and bind to stress response elements. And the activation of these various stress responses results in diminished expression of these genes and G1 arrest, diminished replication stress, and increased longevity. But in our populations, our immobilized cells are being fed excess nutrients. They're seeing an excess of phosphate, an excess of glucose, an excess of nitrogen. Not surprisingly, the direction and the magnitude of these arrows is in the opposite direction from the usual REM15 activation. Okay, so that part of the signal transduction pathway looks like it's performing as it should be. But if you look at REM15, you see that REM15 is highly expressed, even under conditions where you would not expect it to be expressed. And REM15's downstream targets are all appropriately expressed. The cyclins are down, the cells are G1 arrested, and they sure as hell are long-lived in as much as they show less than 5% diminished survivorship over the course of nearly 3 weeks. So our operating hypothesis is that somehow nutrient signaling is decoupled in one or more of these nutrient sensing pathways. And I'd like to emphasize that much of how we realize the awesome power of yeast genetics is by studying mutants. These are not mutant yeast. This is a wild type cell. And these wild type cells are showing regulatory pathways that folks did not realize existed simply because they had not done any genetics in this particular culture condition. So to summarize, I've hoped to have convinced you that these cells show high substrate flux, low rates of reproduction, transcript abundance is up in glycolysis and in the sporulation and cell wall genes, down in the TCA cycle and the cell cycle, REM15 and MSN4, both of which have been implicated in caloric restriction mediated longevity, and that the expression profiles suggest that these cells are arrested. They're undergoing changes reminiscent of sporulation, even though nutrients are in excess and metabolism is active. So I think that we have been successful in this model of uncoupling cell division from metabolism in the absence of caloric restriction. So these cells are long-lived, eating a lot, but behaving in many respects like stationary phase cells. So where do we want to go? We want to determine the cause of REM15 activation. We want to test whether diploids are able to sporulate under these conditions. If we can show that a wild-type diploid can sporulate in the presence of excess nutrients, this would be really a remarkable find. We want to increase bead stability because, frankly, right now we can't push these experiments beyond about 3 weeks because the beads start to break down. This is not a trivial technical problem. And then the money shot, or at least what the NIH has given us money to do, is to test the barcoded yeast knockout collection in the immobilized cell reactor with a view towards discovering new genes having to do with chronological lifespan. The YKO collection is basically a collection of genes. Each of one has a single gene deleted, okay? And these single gene deletants also are coded. They have a molecular barcode that's associated with them. The experiment is pretty simple. It goes like this. We load into the immobilized cell reactor a genetically heterogeneous population of cells, load them into the reactor, and basically turn it on and keep taking cells out and use our barcode microarray to determine the composition of that population. And so we're interested in determining which mutants relative to the wild type are the first ones to drop out of the population, and which deletants are those that are still around at the end of the experiment. And so I will move on and say thank you very much. I appreciate your attention, and hopefully there's time for 1 or 2 questions.
We do have time for a couple of questions.
Yes, ma'am. Or yes, sir.
I was just wondering, does this mean that RIM15, what this shows, that RIM15 is maybe not responsible for longevity like people thought it was, and now you're going to be looking for genes that are possibly responsible for that, or?
I think that RIM15 clearly plays a role, and that RIM15 it's the inappropriate expression of RIM15, which is causing the longevity effect in our system. You know, what's really unexpected is that you have all these different nutrient signaling pathways. I mean, it's a highly redundant system, and usually it never fails. This is the first time this has been observed. And so whether it's, the important thing is this an artifact, of the culture condition, or is this something that yeast have actually evolved, over the millennia? Is this adaptive in some respects? And one of my sort of academic godfathers, and don't quote me on this, David Botstein was saying in his usual expansive way, well, this is the way everybody lives out in nature. That if you find yourself inside, of a grape, or inside of some fruit, or inside an oak tree, places ecologically where you will find yeast, then I don't know whether they're immobilized, but they're certainly growing within a semi-solid matrix. They're not growing on that matrix like they would grow on solid media. They're not growing in that in a matrix that's anything like a dilute cell suspension. So that's an open question. Yes, ma'am.
I guess this is a confusion or big picture question.
Mm-hmm.
So when animals eat too much, they become obese, and normally when yeast and bacteria are eating, they're dividing. So if these yeast, I'm assuming they're still endocytosing nutrients in the matrix.
Oh, yeah.
Where is that going? Are they getting bigger, or?
In fact, they're getting smaller.
How is that?
Well, so the question really is, they're eating a lot, and so where do the nutrients go? One thing that they're doing, and this is, of course, this is why people in the chemical engineering field are so interested in this, they are passing a lot of this out as carbon dioxide and ethanol. So you have these fantastic rates of ethanol production and carbon dioxide evolution. So if you follow the carbons, a lot of it is going there. We also saw that, at least over the limited time course of our experiments, that they're accumulating these enormous reserves of glycogen. And so they are getting not larger, but they are certainly, you have more onboard reserves. The question is, can yeast get smaller, or is cell size variable? Well, the short and simplistic answer is that as they go through the cell cycle, cells have to reach a critical minimum size before they commit to undergoing cell division. And haploid cells and diploid cells are different in size. And you can go out in nature and identify yeasts of slightly different size. I mean, they don't look like they're very different under the microscope, but the difference between an average diameter of, say, 8 and an average diameter of 10, I mean, that's a big deal. So there is some variation, but a lot of it is basically physiological in nature, mediated by the cell cycle or by ploidy. Tetraploid yeast are larger. And so, yes, sir.
Yes, a key difference, if I understand this correctly, is that the immobilized cells are inhibited from reproducing. Does this suggest that it might be an adaptive strategy to hang on until conditions become favorable for reproducing?
That's an interestingly worded question and a good one. So in a sense, the conditions are good right then and there. I mean, they're basically swimming in nutrients. The question, and I can't give you the answer to this, I can only speculate, why are they ceasing to divide? Aside from how the nutrient signaling pathway seems to be different in these cells, we've tested whether just carbon dioxide, so much carbon dioxide is resulting in cell cycle arrest. And one thing I didn't show you guys is that if you take one of these beads and slice it open and look at them for scanning EM, what you see is the yeast have not filled up the bead. They have not filled up the beads. They are not physically restricted from further reproduction. Rather, what you see inside the beads are a galaxy of microcolonies. And so some people have suggested that there's some kind of quorum sensing mechanism, that microcolony size reaches a certain point and then cells stop dividing. But by day 5 and all the way up to day 17, you have no further growth, and you have these microcolonies.
I was curious whether the RNA extraction process is so painful that it would limit the possibility of doing something like a system-wide gene expression profile like Rick Young's lab has done in yeast?
Well, I mean, this is by system-wide, what do you mean?
I mean, as many genes as possible.
That's what we just did.
You're doing, okay.
Yeah, no, that's exactly it. What we haven't done, I mean, as you're probably aware, there are a whole collection of small RNA molecules that certainly have regulatory activity, and the Affymatrix chip that we use certainly is not picking up on those. So.
Thank you very much.